So today our goal is to discuss linkage and recombination, which was the topic, crossing over the topic for the movie to have watched for this morning. And the weird thing about this is that Mendel, we haven't talked about Mendel's P's really. I'm assuming that everybody's heard about Mendel's P's in other classes. We can spend a little bit of time talking about it today, only because his data were too good. And there's a little section we'll cover later on in class from your textbook reading, the OpenStax biology textbook, that claims this. And you might have glossed over that part if you happen to read that part of the chapter. But it's, I think, well worth exploring. Mendel did a number of dihybrid crosses. What were some of the phenotypes he looked at in pea plants? Color of the pea, yellow versus green, shape of the pea, wrinkled versus smooth, round. There were a number of other traits he studied as well, but those are the most famous two: pea color and pea shape. And look at that. It's right there. He made dihybrid combinations of all of those different traits. So there were flower position and flower color, and there were a number of other traits that he looked at. And every time he made, this is directly applicable to the exam you just took, Punnett squares, every time he crossed a selfed dihybrid, like we have up here in the F1 generation, every time he selfed one of these organisms, a dihybrid of any of those phenotypes, he saw that 9:3 to 3:1 phenotype ratio that you expect from a dihybrid cross, the 4x4 Punnett square. And that's really odd, actually. We're going to come back to that by the end of class. That he always saw a 9:3 to 3:1 ratio regardless of which traits he was looking at. That's because linkage suggests that he probably shouldn't have always seen a 9:3 to 3:1 ratio. So the goal for today is to understand how linkage is basically the opposite of independent assortment. So we've got Mendel's second law, independent assortment. That's what we use to set up Punnett squares. You should get equal numbers of different haplotypes of gametes. Big A's with big B's, big A's with little b's, little a's with big B's, little a's with little b's. That's independent assortment, how we set up Punnett squares. Linkage is the opposite. It destroys independent assortment. And it does so through crossing over, which again was the video to have watched today. So important things that we're going to cover for building on for our future classes are going to be nomenclature of linkage, and then also parental and recombinant haplotypes. So those are the key things to be watching out for today as we go through this material. The nomenclature, and then identifying parental and recombinant haplotypes. Let's see how we're doing. So question 1. What does linkage mean? So yes, A is correct. Linkage means that genes are located on the same chromosome. This is the definition of linkage. If I ask you, are these genes linked, and I draw you a chromosome, and both of those genes are on the same piece of DNA, they're linked. So that's good. Has nothing to do with whether genes are transcribed together, although, well, that's an operon if you're thinking bacteria and genes that all get transcribed at the same time on the same transcript. That's linkage, but then bacteria only have one chromosome, so by definition all of their genes are linked. So it does not mean, B is incorrect, it does not mean necessarily that they're transcribed together. Genes do not independently assort. Linkage means that genes are on the same piece of DNA. So what does that mean about inheritance patterns? If two genes are on the same piece of DNA, happens when that chromosome goes into meiosis and then creates another organ. Those genes independently assort. They segregate into different gametes. They're next to each other on the chromosome. They're going to travel together from chromosome into gamete into the future generation.
Assuming no Mendel, right?
Hence, pardon?
Assuming no Mendel occurs?
Assuming no recombination occurs or crossing over, that means that linkage means that genes do not independently assort. Genes are linked. They travel together from generation to generation. So C is another correct answer here. Questions about that? This is just introductory information. Should have gotten it from the movie, but we'll go through it in more detail in class if we need to. All right, nomenclature. Which of those genotypes indicate that the genes are linked? Ah, fantastic. So A is the correct answer, B and C are not. And enough of you got that correct answer that I'm not going to go through this in great detail unless somebody screams and hollers, which I'm happy to go through. Those other two genotypes are kind of tricky. B, what kind of a genotype is B? What do we call that genotype? Big A over little a, semicolon, big B over little b. That's a dihybrid. Right. That colon, the semicolon means what? That two pieces of DNA are on separate chromosomes, hence they're unlinked. The two genes. A is on one set of chromosomes, B is on a separate set of chromosomes. They're unlinked. That other genotype makes no sense at all. A/B. You don't do that. I did it just for fun. It's always A/A, uppercase or lowercase, and B/B. So that third genotype was an effective distractor, it looks like. So linked genes, you write them next to each other with no punctuation in between them. Big A, big B, / little a, little b. Let me show you what that would look like just for completeness if we drew that out. So this genotype, what's it going to look like if I draw chromosomes? Here are four chromosomes, 2 pairs. If gene A is there, where do I put gene B? Below it?
Yeah.
Who says below it? Who says above it? Well, of course, now that I raised that possibility. So we don't know. And that's one of the reasons that geneticists are interested in studying linkage. Linkage helps us learn things about genes like where they are relative to each other on chromosomes. This used to be a really important thing because that was in the days before anybody had the ability to go sequence an entire chromosome and learn all of the nucleotides that make up a chromosome. People still use genetic mapping, that is, using linkage to understand where genes are relative to each other in organisms that are still difficult to sequence the DNA of. So it's still valuable information to have. And just for complete clarity, the reason here that I drew the capital A connected to the capital B is because that's what that nomenclature tells me. The big A and the big B next to each other, no separation, no punctuation between them, means that those are the two alleles that are on the same physical piece of DNA. You could do something like this, big A, little b, / little a, big B, which would look like that. Or the B's could be on top and the A's could be on bottom. Questions, concerns, clarifications needed? Somebody When you have two genes on the same piece of DNA, you just write the allele nomenclature next to each other with no punctuation. Important assumptions about crossover. Okay, everybody randomly jabbed at a letter, more or less. So, this is a difficultly worded question, I admit, that not always true part in the middle is kind of strange. So let's go through this briefly. Does a crossover happen at a random position on a chromosome? So you've got chromosomes running in this direction. Right? In the movie, I just closed my eyes and I stabbed the pen on the glass, and that's where the recombination event happened. That's not, technically, I will never lie to you unless I tell you I'm going to lie to you. So this is me telling you I'm lying to you. It's not always true that crossovers happen at a random position. Most of the time, yes, it's a reasonable assumption to make. But there are some times, there are human chromosomes, for example, where recombination almost always happens at certain positions. There are some chromosomes and some sites on some chromosomes where crossing over happens more frequently than others. But in general, it's a reasonable assumption to start with, that a recombination event will happen randomly somewhere on a chromosome. Part B, they generate genetic diversity by producing new combinations of alleles. That's the point of crossing over in meiosis. But that's always true. So you can decide whether or not that's the right answer or not. Yes, crossovers do generate genetic diversity. It's one of the benefits of having it occur during meiosis. And we'll see an example of that in a minute. Let's see, one crossover happens in each tetrad every meiosis. The top vote getter, yes, that's one of the assumptions. That's also a reasonable assumption to make, but not always true. In humans, multiple crossovers happen between chromosomes every meiosis, but we ignore that because that complicates mathematics. Human geneticists don't ignore it. Human geneticists, and if you want to go more into that, maybe an advanced genetics course is the way to learn more about how you correct for the fact that multiple recombination events happen between chromosomes.
So what was the right answer?
That's C is correct. It's an assumption. It's almost always true. One crossover happens per tetrad per meiosis. And again, just to make that really clear, since that's kind of an abstract concept, and we're going to do this, every one of you is going to do this in a little bit. Crossing over happens during meiosis, right after synthesis. So you get synthesis, you get metaphase of meiosis 1. Where you form, what's this called again? When all four sister chromatids line up together, tetrad. It's a tetrad of the same chromosome. This is where recombination happens. And it involves two chromosomes breaking. So let's say, let me number these, 1, 2, 3, and 4, left to right. Let's say two of these break right in the middle. Chromosome, chromatid 2, and chromatid 4. We have enzymes in our cells that do this, by the way. This is not random, wait, why the hell are my chromosomes breaking during meiosis? This is the cell encodes proteins that actually cause chromosomes to break and recombine. It's that important to generating genetic diversity and to the way meiosis works. And then what happens is those two chromatids will connect to each other. So that, for example, in blue, the top part of chromatid 2 becomes connected to the bottom part of what was chromatid 4. And vice versa. The top part of chromatid 4 then becomes connected to what was the bottom part of chromatid 2. That's crossing over, that's recombination, meiotic recombination. So that's what I mean by when I say one crossover happens per tetrad per meiosis. In reality, other crossovers can also happen like this and like that and like that for every meiosis. So we just simplify things by saying we're just going to look at one crossover per tetrad per meiosis. That would be 4 that I've diagrammed up there. Questions, concerns? So questions, the correct answers there were A and C.
So what were the right answers?
Pardon?
What were the right answers?
A and C. The crossover happens at a random position on a chromosome, good assumption to make. And that one crossover happens per tetrad per every meiosis.
Okay.
Those are the important 2.
By any chance, could you turn off the lights so I can see? Yeah, we can't see.
But you said B is usually on true.
So it depends on which, tell me which looks better. Is that enough? More lights off? Well, okay, if you fall asleep, I'll throw something at you. Surprise, what week is this? You guys have not been seeing in the back for... You gotta tell me these things. But thank you for speaking up about that. Okay, nobody fall asleep. Now, oh goodness, in this cross, which are the parental haplotypes? Hooray! Nicely done. Good job. So that's good. We've been working the last couple of weeks on figuring out haplotypes from genotypes. So that went as intended. And lastly, as I alluded to in the discussion of some of these earlier questions, yes, absolutely, scientists use crossing over information to figure out questions B and C, the top 2 vote getters. We know how far apart 2 genes are. How do we figure that out? It's called genetic mapping. That's what this process is we're going to look at in this chapter.
Where one primer sticks and another primer.
If,
Also sticks.
what were those assumptions?
It's the recombination frequency, so how commonly 2 genes are seen together linked.
If 2 genes, if my arm's in a chromosome, this is one telomere over here, and here's the other telomere. If 2 genes are on my fingers, here's gene A and here's gene B, what's the probability that recombination event is going to happen between them? Pretty high. What were the assumptions we made? What are the assumptions we make about recombinations we just talked about?
Randomness.
Yep, randomness, and that one always happens every tetrad, every meiosis. So if I'm a chromatid, if you're going to, somebody throw something at me. Something soft. Nice try. Okay.
I don't want to hit anyone.
Highlighter. So, let's assume it hit me right here. That's the random sided recombination event. If a recombination event is going to happen somewhere between these 2 genes, where is it going to happen? Somewhere in between them. So genes that are on the ends of chromosomes guarantee you that recombination event, that enzyme that breaks the chromatids, come collect your highlighter. Do you want it now?
You can throw it back.
Thank you. That crossing over event will happen absolutely somewhere between those 2 genes. Now, what if we're talking about the genes that are where my eyes are located? What's the probability that from the back of the classroom she can hit me between the eyes with that highlighter? 0. Pretty low. Let's not test it. We can find out.
We're scientists, we're supposed to test.
That's absolutely right. Null hypothesis and all that. So the closer together genes are, the less likely they are separated by a recombination event. So scientists use the observations of how frequently 2 genes are separated by recombination to tell them something about how far apart they are on the chromosome. Genes at the ends of the chromosomes, every time meiosis happens, will get separated by a recombination event. the closer together two genes get, the less likely it is by chance that that recombination event happens between them and separates them. So that's how C is correct. That's how scientists use recombination to figure out how far apart two genes are. And as was suggested up here in front, that's what we refer to as recombination frequency, which we'll get into next class or the class after that.
I thought that I'd read something about crossing over being used in comparison for expressivity too, for number D.
So which allele, unfortunately no, D is not correct. So crossing over recombination doesn't tell us anything about dominance. But it also, so it tells us not only C, but also B. So B and C. If two genes are on the same chromosome, we can figure that out from recombination, crossing over, and then how far apart two genes are. Here we go with the P's. Sorry, the Apple TV is not working today, which is why I'm stuck up here with a cord and an iPad. Otherwise, I'd be wandering around making sure you're all paying attention and not looking at my YouTube videos, because I know that's what you'd be doing. Okay. So Mendel's P's, mated dihybrid, these are his data. Didn't make these up, real data. Created a purple flowered, long pollened F1 plant here by crossing two. Let's see, what are these genotypes? These are pure breeding genotypes in the parents. They're homozygous at both loci. Okay, capital P is dominant, makes purple flowers. And capital L is dominant, and it makes long pollen. This might sound familiar. I used that, or it will sound familiar if it doesn't yet. I used it in one of the videos you're about to watch. And so we've got a plant that's dominant, homozygous for both dominant alleles on the left side here, and we've got a plant that's homozygous for the recessive alleles. So two little P's, you get red flowers, no purple. Two little L's, you get round pollen, not long pollen. Okay, so make an F1 flower, he selfs it. And if you draw out the Punnett square for this, which we're not going to do in detail, you get a 9:3 to 3:1 phenotype ratio. And we saw that in the past. And that's why down here in the table, that's how they got this expected ratio. So if he looked at, how many observed plants is that? 296 plus 19 plus 27 plus 85. I don't know, somebody do the math while I'm up here yammering away. He looked at this many plants, but he didn't see a 9:3 to 3:1 ratio. What did he see? I calculated it here for you in that blue box or whatever color box that is. What would you call that ratio? The observed ratio. 11 to, sure. So I heard 11 to 1 to 1 to 3, if you round. And that should total 16, 11, 12, 13, 16, right? So both of those ratios total 16, which is the number of squares in that Punnett square, so that's okay. Wait, why didn't he see a 9:3 to 3:1 ratio? Because the Punnett square tells us that's the phenotype ratio we should see when we look at two traits. That's what we've been talking about for the last week and a half or two. Do we have to observe more plants? So it's possible that by chance, those two ratios are the same, we just need to look at more plants. Or it could be that linkage is messing things up. And this was the first evidence that anybody on the planet had that two genes could be located on the same chromosome. Linkage. What are the haplotypes of the gametes of the P0 generation? The purple flower, what sort of haplotypes does it produce in its gametes? Well,
Big P, big L.
thank you. PL, that's a cheating answer. Okay, not cheating, but Yes, absolutely right. PL, oh, no dot there. We don't know if they're linked or not, but here's the punchline, they're linked. And the other plant produces little p, little l gametes. Okay. The gametes that the P0 generation individuals produce are parental type haplotypes, by definition. The P0 generation, not the F1 generation. Critical distinction. Those are the parental haplotypes, because they're the haplotypes of the parental P0 generation. So in contrast, there are two parental haplotypes, and there are two recombinant haplotypes. What are those? What are the different combinations that you could have if independent assortment was occurring? You'd get other gametes that have, what? Yeah.
Capital P and little l,
Yep. Capital P and little l,
or little p and capital L.
and little p with capital L are the recombinant types. This is just like independent assortment. It's just like setting up a dihybrid Punnett square. You get 4 different types of gametes. Big P with big L, big P with little p with big L, and little p with little l. 1, 2, 3, 4. Any questions, concerns so far? Yeah, please.
The two middle ones, the ones with the really low ratio,
Yes.
those ones that happen to get hit between the eyes.
Ah, indeed. So that's a good way of putting it. We're going to see that in just a second. But these, you'll note, the point was, we expected to see a larger number of those phenotypes than we observed. There are 2 phenotypes. This is the secret to detecting linkage. 2 phenotypes you see more of than you expect. 2 phenotypes you see less of than you expect. So the ones that are in boxes here down in the bottom table are the 2 phenotypes we saw fewer of than were expected. We thought we were going to see 80 of each if there was a 9:3 to 3:1 ratio of the 4 different phenotypes. But instead we saw 296, 19, 27, and 85. So the point of that slide was just to go over the parental versus recombinant haplotype identification. Look at the gametes that are produced by the parents. Those are the parental types. The other 2 types in dihybrid would be the recombinant types. Saw one other hand up. Okay. Finally, everybody said together. I'm going to send you this Google Classroom assignment. Yeah. Here's what it looks like. And you're all going to get this right now. Right on time. So this is what it looks like. And it's got some instructions on it. I'd like you to follow the instructions, work on this by yourself, talk to a neighbor if you like for about 5 minutes. Before you do that though, everybody close your eyes. Close your eyes. Or at least don't look at your tablets. Pay attention to me. I'd like you to pick 2 numbers between 1 and 4.
3.
Everybody pick 2. You don't have to tell them to me. So inclusive, 1, 2, 3, or 4. Pick any 2 of those numbers, 1, 2, 3, 4. Pick 2. Okay. And then pick a letter, A, B, C, or D. So you should have 2 numbers in mind, 1, 2, 3, or 4, 2 of them, and a letter, A, B, C, or D. Okay? Easy, right? The easiest thing you had to do as an undergraduate? Okay. Now, the point of this exercise is the 2 numbers you picked are going to be the 2 chromatids that are going to undergo crossing over. I've numbered them down at the bottom there. 1, 2, 3, 4, left to right. And if you picked letter A, what you're going to do, so let's say you picked numbers 1 and 3 and letter A. I want you to draw that crossing over event happening beneath the letter you picked. So here I drop between A and B. And between the two numbered chromatids that you picked. So the hope is that everybody chose some random chromatids and random letters, so we're going to get a good distribution in a class this size of where recombination happens and which chromatids it involved. Then the only goal, main goal, is in these bottom 4 blanks, write out the 4 different haplotypes of the 4 different chromatids. So in this case, I would write out, I picked up a capital A there for chromatid 1. I had a capital A, and then there was a recombination event. So then I get the rest of the letters from that chromatid. Capital B, lowercase c, lowercase d, capital E. Okay. So you work on this for about 5, 6, 7 minutes. Raise your hand if you've got questions about how to do this, but that's the idea. Draw the crossing over and then write out the haplotypes of the 4 different chromatids after recombination occurs.
Yeah, so I think there's
Because that's the possible combination, because essentially they are alleles, but the little combination.
Yes, exactly. That's what happens. Yeah, random, wherever possible.
Well, at least
So this was just exercise. And when you're done with that, please attach it back to the assignment and send it back to me by submitting when you get there. Questions? We're going to use these data next class, so thanks for helping me collect these data. Yeah.
Once you cross, does everything of the underneath it follow?
Right.
2 normal ones?
Yes.
Those are backwards, so.
What would be the plan? So you got capital A, that one links up down here, so then you start writing capital A, capital B, C, D, E, that's here. Lowercase a,
So that crosses over again. The chromosomes will break
Think how I
between
chose
B
C for
and
this
C.
one, exactly like
And
that,
then
capital
you
B.
get little a and capital B, and then that
Wait,
part
would
of
the
that
whole
goes
bottom
over
section of
over
it be
here
transferred
to
over?
capital C, capital D, lowercase.
I think we're just looking at allele itself, not
And
the
the
entire,
top,
not the
that
entire
next to
segment.
the bottom.
Yeah.
Yep.
Because if that was the case, then we'd actually transfer all this over here, and that's not what we look at.
Yeah, you just have to add a.
That's not the example he gave us.
So those,
He only did one, he only did one and not 2.
Okay. So, a couple of you are realizing that, what do you write in the other 2 blanks? You've got 2 chromatids that break and cross over. What happened to the other 2? They don't do anything. They didn't break. They didn't cross over. They didn't recombine. So every meiosis, if there's one recombination event, that means that there are 2 chromatids that are what? They've broken, they've exchanged arms. That's called what? Starts with an R. Recombination. Those are recombinant haplotypes. What are the other two? Those are parental types. They don't break, they don't recombine, they don't exchange bits. So you're going to wind up generating 2 recombinant haplotypes and 2 parental haplotypes every meiosis. So if I finished filling this out in my example, I would have that. Those are 2 recombinant types. And the 2 chromatids that didn't do any crossing over stay the same. Nothing changes. Those are parental types. So those gametes that get produced with those chromosomes in them will create offspring that are parental type offspring. You see that in the questions? Yeah.
I was just wondering, so you said you could pick any chromosome between 1 and chromatid between 1 and 4.
Right.
So wouldn't, let's say we pick 1 and 2, wouldn't recombinant just result in the same thing?
That's a very good point. So I hope everybody is paying attention. Thank you again. Sorry to cut off conversations that are on topic, but just for a second. So what happens if you choose chromatids 1 and 2 or chromatids 3 and 4 to do recombination? So did anybody do that? Pick 1 and 2 or 3 and 4. So what happens? When recombination happens between, so let's say I picked 1 and 2 and the letter B. So crossing over occurs between B and C. Nothing's changed. You get capital A and lowercase b with capital C, capital D, and lowercase e, which is exactly the same as the other crossover product. So in this case, you get parental type, parental type, parental type, parental type. Because even though crossing over happened, if crossing over happens between sister chromatids, right? Chromosome 1p, the nomenclature down on the bottom I didn't describe, 1p, what does that say? 1pn and 1po, 1 paternal. So if those are the 2 copies of the paternal chromosome, they're genetically identical. If crossing over happens between them, we never see it because it doesn't make new combinations of alleles. Crossing over has to happen between paternal and maternal chromatids to make new combinations of moms and dads alleles. So if you picked 1 and 2, or if you picked 3 and 4, it doesn't matter where recombination happens, you get 4 parental types. This is going to be really important later when we talk next class about the limits of recombination. What's the most recombination we would observe and the least amount of recombination we observe? This is an example if you choose to have crossover happen between sister chromatids from the same parent of the least recombination. We don't observe any recombination. It happened, crossing over happened, the chromosomes broke, exchanged arms or exchanged pieces, but as a scientist, we can't tell that that happened. Because there are no new combinations of alleles.
You said we're not going to look at it, but there are multiple recombination events going on, right?
Yes.
So do these chromosomes just kind of break up and then all come together?
So in reality, if this is a human chromosome, for example, you'd also potentially have a crossing over event happen there at the same time. So you'd have the first crossover event that doesn't change anything about the haplotypes, but the second one would, because it's involving 2 non-sister chromatids, one from dad and one from mom. And then things get complicated pretty quickly, as I hope you at least start to appreciate. So please send those documents to me somehow when you're done filling them out. Doing the crossing over, writing down the 4 haplotypes you generated, please send it to me. Those are important data for helping with class next time. Thanks for participating in that. So to wind up, move us into the next class over the next 8 minutes or so. Here's that section from the textbook I was alluding to earlier about how Mendel's data seemed to be too good. People are starting to wonder, did Mendel actually falsify his data? Did he commit scientific fraud? Those monks, fraudsters. So researchers have mapped the 7 traits. Here's the important part. They mapped the 7 traits onto the 7 chromosomes of the pea plant genome. So wait, he had 7 traits. This is odd. You look at 7 traits, and there are 7 chromosomes in peas. And was Mendel enormously lucky to select only unlinked genes, or as it says later down, did he find some evidence of linkage and then throw it out? So we can do a little bit of genetic forensics, go back in time and try to figure out, did Mendel falsify his data? Did he not report all of his data? Did he, it doesn't really matter because ultimately he did come up with Mendel's laws, which are true, but the way he did it, I don't know. So those are the 7 different traits that he studied. We've talked about a couple of these already. So there's 7 different traits, and we can skip these. A lot of people, including some famous geneticists, have written on this topic. That's those 2 slides, which I'm skipping over quickly. So he used those 9:3 to 3:1 ratios to infer that all of those 7 genes controlling 7 different traits were independently assorted, which hopefully by the end of this chapter in your minds, every time I say independent assortment, or anytime one of you says independent assortment, you should think independent assortment means separate chromosomes, 2 genes on 2 different chromosomes. That's how independent assortment works. He actually picked and chose which data he reported. So ultimately, here are physical maps, meaning that the lengths of each of these are related somehow to physical distance. How do we measure physical distance in genetics? The physical length of a chromosome. Yeah. Physical, so we're going to compare, in this chapter, 2 different types of maps. There are physical maps of chromosomes and there are genetic maps of chromosomes. What physical unit can we use to measure how long a chromosome is? Millimeters?
Micron?
Well, we could, but what's the physical unit that makes up a molecule of DNA, a chromosome? A base pair. Yeah, so we use base pairs as the measure of a physical length of a chromosome. Right. So human chromosome 1 is 100,300,091 nucleotides long. I made that up, don't quote me. So in this map, they're showing us the location of lots of different genes in the pea plant based on how far apart they are in physical distance, in numbers of base pairs. Everything is relative. What do you notice about the locations of the genes that control those 7 traits? They're on different chromosomes. There are 7 traits and 7 chromosomes. Where does that generalization break down? There's one case where that's not true.
Mostly in the middle.
Right, so the 3rd chromosome, pod color is in the middle somewhere, and height is down there at the bottom, and that means that there's nothing on chromosome 6. Right? None of the traits that Mendel studied had genes controlling them that were located on chromosome 6. And there were 2 genes relatively far apart, importantly, on chromosome 3. So what's the chance that Mendel randomly picked 7 different traits to study, and 6 of those 7 were distributed evenly across the 7 different chromosomes of peas, and the 2 traits that were on the same chromosome are relatively far, controlled by genes relatively far apart on that chromosome. That's what all of these points, including your textbook and all of these papers that geneticists have written, have argued. That would be exceedingly unlikely by chance to have happened. And that Mendel must have known, he must have figured out that linkage was occurring between traits. And so maybe he didn't show us those data. Well, that's weird, those data don't fit the 9 to 3 to 3 to 1 ratio. Burn that diary, or that lab notebook, right? So nobody knows. He only showed us the data that conformed to his assumption about how genetics was working. Every time he saw a 9 to 3 to 3 to 1 ratio, maybe, no one's proven this, we can't, maybe those are the data we have of his, the ones that fit his expectation. Like I said, regardless, he still figured out how linkage works. Well, he still observed linkage and independent assortment. So the one place he might have seen linkage is what? Pod color and height. Okay, so there's one cross that he did where he might have seen a little bit less than a 9 to 3 to 3 to 1 ratio, something different. But in general, this is so, winding up. Here's the take-home message for today that will move us straight into class next time, and there are a couple other slides after this. Really quick. So if you have two genes that are on separate chromosomes, they will independently assort. You get that 9 to 3 to 3 to 1 ratio if you're a dihybrid. If they're on the same chromosome, you expect something different. If the genes are linked, you expect to see something other than a 9 to 3 to 3 to 1 ratio. So what we do at this bottom point, when you see something that's not a 9 to 3 to 3 to 1 ratio, that's when you think, those genes might be linked. They're not independently assorting. And that is, in fact, what was happening in this case. So we see what was termed earlier an 11 to 1 to 1 to 3 ratio. The question to you for next class, because next class we're going to start talking statistics, which was a point that was sort of marginally raised over here earlier, was, are those observed and expected numbers really different, or are they just different by chance? So if we look at more p's, would that 11 to 1 to 1 to 3 ratio eventually become a 9 to 3 to 3 to 1 ratio? If we look at an infinite number of offspring. We can't look at an infinite number of offspring. So we're going to have to do some statistical analysis to figure out if those two sets of data are really different. So, we got one new textbook to download for next class. If you haven't already, statistics. The only chapter we're going to read in that textbook, free online. Chapter 11, just one section, section 2. And then watch the chi-square test movie. And we'll come back and we'll answer that question about whether or not those ratios really are different or if they're the same. Are the genes linked or do they independently assort?